We got a last minute question. We'll go ahead and take this. Frankie, if you're still available. The
Yep.
comment is wonderful talk. And then the question is, could you comment on the neuronal projections of these AGRP neurons that contribute to changes in locomotion?
No, I can't. That's a really good question. It's not something that's been heavily investigated. I'm not entirely sure if it's signaling to MC4R downstream receptors, maybe neuronal population in the PVN. I'm not entirely sure. I don't. There are lots of possibilities, but it's something that needs to be investigated further. It's a great question, though.
Great. Thank you, Frankie. Okay, now we can go ahead and move on to Janae Vacharasan. Janae, if you wanted to go ahead. Oh, okay. Sorry about that. And Janae is pre-recorded. Excuse me. Here we go.
Hi, I'm Janae Vacharasan, and today I'm excited to talk about my work with the role of IL-17A and the etiology of autism. So what is autism spectrum disorder, or ASD? It's a group of behaviorally heterogeneous disorders characterized by impairments in social interaction, communication, sensitivities to certain stimuli, and repetitive behaviors. So what we have learned is that autism can arise from copy number variations, single gene disorders, and chromosomal abnormalities. But a lot of our understanding of autism is still unknown. From rodent studies, one hypothesis is that ASD arises from maternal immune activation. And this diagram here helps to show that when the mother has a severe infection, for example, she can activate a subset of lymphocytes that lead to the production of IL-17, which can cross the placenta and affect the fetal brain. So many maternal immune activation studies have been done in rodents, but there has been a push to look at human cellular models since in rodents, full ASD behaviors do not fully phenotypically produce in rodents. So in our lab, we've created an environment in human stem cell-derived neurons. Here in the first part is our dual SMAD inhibition protocol and how the timing maps in vivo. In this experiment, I conducted what is similar to a critical period experiment for rodent labs, where I treated cells with IL-17A at different time points in the process of neurogenesis, and then measured their synaptic activity at day 55 and day 60 using a multi-electrode array system. So day 1, they are still mostly embryonic stem cells. At day 12, they are forming their neural epithelial sheets, and at day 17, they are neuronal progenitor cells organizing themselves into rosettes before they become cortical neurons. So what we can see in the quantification here and visually by the activity map below is that IL-17A treatment decreases the synaptic activity, and early exposures seem to decrease the synaptic activity even further. Next, we found that IL-17A significantly decreases the neurite length, and moreover, it increases the neuronal production of other cytokines such as interferon gamma and IL-13, and this was measured by the Luminex cytokine array, and these results were different than our positive control lipopolysaccharide. So overall, IL-17 seems to be decreasing the neurite length and inducing other cytokines. Moreover, we ran qPCR on a set of known ASD risk genes and found upregulation in KAT-NAL2 and DYRK1A. So, we see that IL-17A leads to different phenotypes, but could there be a chemical to help alleviate these outcomes? We looked into resveratrol, a potential anti-inflammatory compound that interacts with other cytokines, and found that it rescues the mean neurite length phenotype that we were seeing earlier when undergoing IL-17 treatment. So, in the future, we plan to continue the resveratrol rescue experiments. Additionally, we have some preliminary ATAC-seq, so note this preliminary needs to be repeated, but it showed that the differentially significantly expressed genes were a kinase and genes involved in Golgi trafficking and exosomes. Moreover, we plan to look at the microglial neuronal IL-17 interaction, since we have a protocol in the lab that can convert human embryonic stem cells to microglia. Additionally, we plan to conduct RNA-seq in order to further characterize the differentially expressed genes when neurons have undergone IL-17 treatment. So I would like to thank the members of the Lizarraga Lab, especially Evelyn, Foster, Michaela, Mary Kate, and Penkosh that specifically worked on this project. And I would like to thank our funding sources. And additionally, I'm excited to be here and thank you for having me.
Thank you so much, Janae.
Hi.
Next up, we'll have Crystal Smith.
Hi, my name is Crystal Smith. I'm a fourth-year PhD candidate in the Robinson Lab at the Medical University of South Carolina. And the title of my talk is Understanding the Role of the Insulin Receptor During Transport and Signaling in Post-Stroke Cognitive Impairment. Looking at these maps, when it comes to diabetes, obesity, and stroke deaths, we see increased prevalence in the Southeast region. That is of particular importance to me because I live in the Southeast region. Comorbidities such as diabetes, obesity, insulin resistance, and hyperlipidemia increase the risk of stroke and show increased prevalence in African Americans. Insulin resistance is characterized as hyperglycemia and hyperinsulinemia and is associated with increased circulating insulin in the periphery. So why is this research important? Well, there is a critical need to improve stroke therapies. Currently, what we know is patients with diabetes, obesity, and insulin resistance have worse outcome following stroke. Insulin resistance is characterized as hyperglycemia and hyperinsulinemia and is associated with increased circulating insulin in the periphery and decreased circulating insulin in the brain. A lot of people may not know is the impact brain insulin has. Brain insulin is involved with enhancing memory, regulating food intake and weight gain, is involved with neuroplasticity, myelination, neurogenesis, synaptogenesis, as well as being anti-inflammatory, antithrombotic, and antivasculatory. So knowing all the positive things that we know about brain insulin, this led me to my hypothesis, which is increasing brain insulin levels will improve functional stroke recovery. So first, we wanted to create our brain insulin deficient model. This was done by placing the animals on a standard or high-fat diet for 24 weeks. 6 weeks of diet, we saw that our high-fat diet animals had decreased relative hippocampal insulin levels compared to our standard diet animals. So we have created our decreased insulin in the CNS. Next, we wanted to look at our plasma insulin levels, and we saw that our high-fat diet animals had increased plasma insulin levels compared to our standard diet animals. So we have our increased circulating insulin in the periphery. We have established our insulin-resistant animal model. Next, we wanted to see if any of our results were going to be confounded by our high-fat diet. So we wanted to establish brain insulin deficiency pharmacologically through the use of the insulin affibody. The insulin affibody binds to the insulin, so it cannot bind to the insulin receptor and cannot be transported into the brain. During normal insulin signaling, the insulin binds to the insulin receptor at the plasma membrane, which then initiates autophosphorylation of the insulin receptor. Once the receptor has been phosphorylated, the tyrosine residue is phosphorylated on the insulin receptor docking protein, or IRS1. That phosphorylation leads to activation of AKT, which results in normal functional insulin signaling. However, during impaired insulin signaling, the serine residue is phosphorylated on the IRS1 docking protein, which leads to inactivation of AKT and non-functional insulin signaling. So we wanted to determine which concentration of the affibody would decrease insulin receptor signaling the most. We saw that 10 micrograms of the insulin affibody reduced insulin receptor signaling the most when looking at PAKT insulin receptor respection, which is a measure of our AKT activation. So in conjunction with looking at the affibody's impact on insulin receptor signaling, we wanted to determine the impact it had on cognition. This was done by our animals performing our novel object recognition task. The object of this task is to place the animal in the middle of the arena with two similar objects on either side. The animal is allowed to explore for 10 minutes. 10 minutes following the 1 hour following the exploration phase, a new object is replaced on the right side of the arena and the animal is allowed to explore again. What we're seeing is 5 micrograms of the Affibody is trending towards having an impact on the animal's exploration time with the novel object. So in summary, I've shown that our high-fat diet mouse model is a valid model of hyperinsulinemia. I've also shown the impact of intranasal insulin Affibody on insulin receptor signaling and cognition. In the future, we want to explore how the insulin is crossing the blood-brain barrier. We also wanted to determine the necessity of the insulin receptor through the use of our insulin receptor knockout transgenic mouse line, and also exploring the impact of increasing brain insulin on cognition. I would like to thank my mentors, my lab mates, and all of my funding sources, as well as Black in Neuro, for allowing me to present today.
Thank you so much, Crystal. Next up, we have another pre-recorded talk from Tia Donaldson.
Hello, everyone. My name is Tia Donaldson, and I'm a third-year PhD student in the psychology department at the University of New Mexico. Today, I will be talking about head direction cells and how they differ across the thalamo-parahippocampal circuit. Head direction cells are neurons that fire in relation to an animal's heading within the environment along the horizontal plane. This cell activity is independent of an animal's behavior or location. Head direction cells fire much like a compass, with firing occurring within a given direction. Head direction cells are also thought to underlie an animal's sense of spatial orientation. These neurons can be found within several brain areas known as the thalamo-parahippocampal circuit. These areas include the postrhinal cortex, the medial interrhinal cortex, the postsubiculum, the parasubiculum, and the anterior dorsal nucleus of the thalamus. Previous research has shown that these cells have distinct firing characteristics. However, research is limited on the individual differences. Therefore, the purpose of the current study was to conduct a comparative analysis of these head direction cell firing characteristics across brain regions. We used 39 female Long Evans rats and implanted them with movable microdrive tetrodes and stereotrodes in each of these brain regions. You can see the breakdown of the rats in each group here. Once recovered, rats openly forage for food pellets in a cylindrical arena while the electrodes recorded head direction cells. So getting to the results, these are photomicrographs of the brain areas stained with crescent violet to confirm electrode placement. Below, you can see that we recorded from a total of 734 cells throughout the circuit, with the individual breakdown in each brain region shown below. Given that I'm limited on time, I'm only going to present the results from one measure. Head direction score, or mean vector length, is a robustly directional measure that ranges from 0 to 1. The closer the data is to 1 indicates strong directional tuning. However, if it's closer to 0, that means that the cell activity is more coarsely modulated. Using a non-parametric ANOVA and pairwise comparisons, we determined that the ADN had significantly higher head direction score compared to other brain regions, and this was significant at p.05. In contrast, cells within the postrhinal cortex had the lowest head direction score compared to other brain regions, and this was significant at p.001. So to summarize, we found differences in head direction cell firing characteristics. Cells within the anterior dorsal nucleus of the thalamus had the sharpest directional tuning, while those in the postrhinal cortex had a broader range of firing. These results suggest that cells within this circuit may contribute something unique to spatial orientation. However, further work will be needed to understand these contributions. This research also has implications for Alzheimer's disease because one of the hallmark symptoms of the disease is a decreased sense of direction, and this circuit is also sensitive to Alzheimer's disease pathology. So this is also further work that we are interested in. And with that, I would like to thank everyone in the audience for listening today. Special thanks to everyone in the Black and Neuro team for this incredible opportunity. I'd like to thank my PI, Dr. Clark, and also everyone in Dr. Jeff Tabe's lab at Dartmouth. Thank you for your time and attention, and I will now take any questions.
Thank you so much. All right, next up, we have Gladys Shaw.
Hi, my name is Gladys Shaw, and I'm a PhD candidate at Virginia Commonwealth University. I'll be presenting some data that was recently published, and you can read the paper if you scan this QR code up above. Chronic stress, especially during the unique developmental timeframe of adolescence and early adulthood, dysregulates the hypothalamic pituitary adrenal axis, causing an increase in systemic inflammation, resulting in an increased risk of the development of neurodegenerative disorders. Moreover, traumatic stressors such as predation stress during this time frame may give rise to immune and neurological susceptibility that does not emerge until later in life. The relationship between trauma and metabolic activity is highly translatable, with increased traumatic experiences giving rise to a shift in metabolic and mitochondrial function. This not only damages neuronal metabolism, but it also can lead to increased rates of apoptotic signaling in specific regions of the brain. Data from human trauma studies have shown a similar outcome, with altered mitochondrial function in male war veterans with PTSD. So this leads to our overarching experimental question. Does a combination of traumatic stress during adolescence and chronic inflammation in adulthood induce altered synaptic respiration reflective of these behavioral changes? Shown here is the experimental design, and for the sake of time, I'm not going to go over this, but just know that the terminal endpoints were taken 10 weeks after the last exposure to the stressor and 4 weeks after the final treatment injection. And these animals were given chronic LPS injections for a total of 8 consecutive weeks. From these sex-specific behavioral changes, we wanted to determine the origin of these changes. We chose to focus on the synapse, as it is the site of information transfer between neurons. More specifically, we isolated synaptosomes, a functional unit of the neuron comprised of the pre- and postsynaptic terminals. The coolest thing about synaptosomes is that they maintain full energetic function and neurotransmission, which allows us to probe synaptic bioenergetics in real time. To do this, we utilize Agilent's Seahorse technology and their cell mitostress tests to assess changes in synaptic metabolism, a potential cause to the differential behavioral outcomes that we saw earlier. So looking at this data, in males, a combined history of stress and inflammation decreases overall oxygen consumption in synaptosomal mitochondria, which is not driven by a particular aspect in the respiratory chain assessed. In females, a combined history of stress and inflammation decreases overall oxygen consumption. This is in part driven by the significant decrease in proton leak between stressed females and a history of inflammation versus stress alone. Conversely, chronic inflammation alone increases overall respiration when compared to non-stress saline-treated females. In males, there is no significant difference in relative number of mitochondria, presynaptic terminals, or number of mitochondria within each presynaptic terminal, as determined by Western blots, shown in Figure A. Although there is no significant difference in mitochondrial number in females, results display a treatment-specific difference in presynaptic terminal number, and ratio of mitochondria per presynaptic terminal, as shown in B, with the LPS-treated females, displaying an increase and decrease respectively. In figure C, we see flaming scores, which is an assessment of mitochondrial phenotype and a proxy of mitochondrial health. In the males, there is no significant difference between mitochondrial phenotype distribution, yet within the females, we see that those with a history of inflammation possess lower levels of fully ruptured mitochondria, which is a score of 1, yet they have more damaged mitochondria, showing decreased amounts of swollen or agranular mitochondria when compared to their non-stress treatment match controls. So that's looking at 3 and 4 in the scoring system. So we see that there is a sex-specific difference in transcript levels of TNF alpha and IL-1 beta when looking at qPCR data. So we see that males with a history of inflammation show a significant increase in TNF alpha and IL-1 beta within their hippocampus, yet this difference is not seen within the females. Additionally, within males, within their stress match controls, we see that stress alone increases TNF alpha in the hippocampus. That's also not reflected in the females. Data from the prefrontal cortex shows no significant differences when looking at males or females. Chronic inflammation has also shown to increase circulating ROMO1 in males. So males with a history of inflammation display an increase in circulating ROMO1, which is a marker of reactive oxygen species, or another marker of mitochondrial health, when compared to their saline-treated counterparts, regardless of their stress history. Alternatively, there is no significant difference in the females when looking at either stress differences or inflammation differences. So taken together, chronic predation stress induces long-lasting anxiety-like behaviors, which is attenuated by chronic inflammation in both male and female mice with a history of stress. The changes in synaptosomal respiration suggest that sex, stress history, and inflammation interact to create a unique, individualized metabolic profile. Yet, despite this, there is no significant difference in mitochondrial phenotype or synaptic composition in males, but there is a significant difference in females measured over 10 weeks after the last stressor and over 4 weeks after the final treatment injection. Although peripheral inflammatory markers between the sexes are similar, transcript levels of inflammatory cytokines are heightened in the male hippocampus in a stress and treatment-specific manner, but not in females. These data suggest that the effects of stress and inflammation manifest centrally in females, but peripherally in males. And collectively, these data highlight how sex, stress history, and inflammation can interact to shape individualized patterns of behavior, possibly driven by these altered mitochondrial functions. So I'd like to thank everyone in my lab, VCU's Anatomy and Neurobiology Department, the microscope core that I use to get these images and this data, and Black and Neuro for hosting this conference.
Thank you so much, Gladys. Next up, we will have Jasmine Hendy. And a reminder for everyone who's just joining us, I see we have some new people. If you have questions for any of our speakers, if you could just drop them directly into the Q&A and just remember to indicate who they're for, then we'll get to them during the Q&A session. And it looks like Jasmine may not have joined us yet. So we can go ahead and proceed with the Q&A. So if you have questions, remember, go ahead and drop them in the chat and I'll go ahead and start the Q&A. So this question is for, oh, I'm so sorry. Jasmine is here. Jasmine, if you want to go ahead. Oh, I'm so sorry. This was a pre-recorded talk. My apologies. Jasmine was here and we are ready to go. Here you go.
Hello, my name is Jasmine Hendy from Delaware State University, and I am presenting circuitry and transcriptional modifications trace the expansion of the frontal cortex in humans. In our lab, the question that we asked is what transcriptional and circuitry changes have accompanied the expansion of the frontal cortex in humans? And we focused on 2 points, and the first one is comparing gene expression across the frontal cortex and looking at specific genes called superganglion genes found in the layers of the frontal cortex, as well as integration of anatomy and development, which corresponding ages across species with the use of time mapping. And what you see here is an example of these superganglion-rich genes. And superganglion-rich genes are genes that are present in layer 3 of the frontal cortex. There are 19 comparable genes in the frontal cortex between mice and in humans. Out of the 19, we looked at 4, which are CRIM, SITE2, VAMP1, and AFH. What you see here is an example of one of the superganglion-rich genes, VAMP1, and we quantify the expression of the gene in the upper versus lower layers of the frontal cortex in humans versus mice. Secondly, we're looking at the integration of anatomy and development, which is what you see here. We measure the white matter volume of the frontal cortex over the course of postnatal development in mice and in humans. We use a software called ImageJ where we measure the white matter to compare the expansion of the frontal cortex of humans relative to mice in development. FA is a measure of connectivity in the brain and reflects the myelination in white matter. We're able to study the developmental time course of the supergranular-rich genes with a time translating model, which is what I'll explain more in detail later in the presentation. Here we look at the increased expression of supergranular-rich genes in humans compared with mice. And here we're able to see the supergranular-rich genes in layer 3 across the frontal cortex of humans versus mice, and how humans have an expansion of layer 2 to 3 as well as an increased expansion of supergranular-rich genes in layer 2 to 3 compared with mice. Each graph shows the genes that we looked at and how the layer thickness is expanded in humans compared with mice, as well as the scale beside the graph showing the intensity of gene expression in mice versus humans. Humans have an expansion of layer 2 to 3 as well as an increased expansion of supergranular genes in layers 2 to 3 compared with mice, as you can see here in each graph for each gene that we looked at. Next, we look at the growth of the cortex and how it grows for an unusually long time in humans. And what we use is the translating time model seen here. And results show that over the course of development, the human frontal cortex ceases to grow much later in humans. And we use a times translating model through translatingtime.org to map corresponding ages between humans and mice. For the humans, which is what you see right here, the white matter ceases to grow at 5.5 years of age, which is the top graph, while in mice it ceases to grow 20 days after birth, which is in the bottom graph seen here. On the y-axis, we see the area of white matter in comparison to the days after conception on the x-axis. To conclude, we studied the growth of the frontal cortex and how it is protracted in humans relative to mice, as well as integrating RNA sequencing with neuroimaging level phenotypes as an effective strategy to assess deviations and development across species. We would like to thank Dr. Melissa Harrington and Dr. Maury for the FA scan, as well as thanking Delaware Embry program for funding. If you would like to learn more about our research, you can go to www.charveylab.com. Thank you.
Thank you so much to Jasmine Hendy, and thank you to all of our speakers for that session. Next up, we'll have a Q&A for all of the speakers, with the exception of Tia Donaldson. So if you could drop your questions in the chat. This first question is for Crystal. They said, great talk, Crystal. And they asked, based on your talk, I can understand that insulin is important for memory. But other than that, is there a particular reason why you chose to take samples of hippocampal tissue in the context of studying stroke?
Yes, thank you for your question. So we want to look at cognitive impairment and stroke recovery in that context. So oftentimes patients have memory deficits. So eventually I will be looking at these cognitive deficits in hippocampal tissue in stroke patients. But first we wanted to look at these things in standard diet non-stroke mice to get like a baseline.
Thank you so much, Crystal. Also, look at you in lab.
Yeah.
Working overtime. Thank you so much. Okay, our next question is for Janae. Someone said, great talk. And they asked, do we know the dosage/amount of IL-17 from the maternal side in vitro required to elicit the inflammatory response in fetal neurons/human stem cells?
So that question is currently being investigated, but there was a study done from a lab in Saudi Arabia that measured it in serum levels, and it kind of showed a range depending on the severity of autism. So in the serum levels, some was as low as like 10 picograms per milliliters, but on the upper end, it went up to around like 40 picometers or so. But when you translate that to cells, the lowest amount that we've seen and previous papers have seen that still activates the IL-17 receptor in the neurons would be 10 nanograms per microliters, and 100 nanograms per microliter is like the level at the maximum amount before you begin to see like changes in cell death numbers. So it's still currently being investigated, but it depends on if you're measuring at the serum levels. Those are typically in the picogram range, but if you're measuring from inside the brain or in the cellular actual like neurons, that's in the 10 to 100 nanograms per microliter range. Thank you.
Thank you, Janae. Next up, we have a question from Crystal. Our person said, nice talk. They also said, thanks for explaining your research so clearly. Are there any known differences in insulin signaling across the hippocampus, i.e. the dorsal versus ventral hippocampus and ventral hippocampus?
I don't currently know any, but that is one aspect of my research that we wanted to examine, is looking at differences in the hippocampus. But because it's so small, oftentimes when we're doing takedowns and collecting the tissue, it's hard to really dissect out the different areas clearly. But that is something that we're interested in looking at.
Thank you, Crystal. Our next question is for Gladys. They asked, do you have any thoughts on why there were differences between the PFC and the hippocampus?
So I also was surprised not to see any differences in the PFC following this stressor, since it was so severe and this inflammatory profile. What I'm thinking might be the case is that I'm just guessing that it wasn't salient enough to have like feedback to the PFC. So our main priority with this was to dysregulate the HPA axis and looking at either the hippocampus or the amygdala. And since that does have some feedback to the PFC, I would assume that it would have to have high enough salience of the stressor and the inflammatory profile to show results there. Additionally, another lab member has done some work looking at microglial activation following a similar stressor. And we do see microglial activation in the hippocampus, but not anywhere else in the brain yet. So it might be related to that, but a more exact answer, I'm not quite sure why we don't see anything in the PFC.
Thanks, Gladys. We also had another question for you. This comment first was, nice talk and congrats on the paper. They asked, have you also looked at what happens with synaptosomal respiration in the amygdala of these animals in response to stress? If not, could you comment on that?
So we have not looked at synaptosomal respiration in the amygdala. We just got region-specific synaptosomal respiration to work literally last week. So the data that I've shown here was in the entire brain. So we have not looked at the amygdala. We have looked at hippocampus, and we're looking at PFC this week. So I'm not quite sure what will happen because the hippocampal results were kind of surprising as well. But I'm sure I will post it on Twitter once we figure out what exactly is happening in the regions of the brain. Sorry, I couldn't answer that more specifically.
Gladys, we actually have one more question for you, and this will be the last one before we move on. This person said, great talk, and they mentioned that they've seen some studies showing estrogen-mediated differences in CNS inflammation. Do you think this could apply to the sex differences that you see in your research, or do you have a different hypothesis for underlying mechanisms?
1000%. That is actually the topic of my dissertation research. So estrogen, as well as the other sex steroid hormones, testosterone and progesterone, do have receptors within the mitochondria, and they activate certain feedback mechanisms between the nucleus and the mitochondria to drive transcription of nuclear and mitochondrial DNA. So I do think that either estrogen is driving this or testosterone is not driving this alternatively. To show these sex-specific differences. I haven't done deep enough research to figure out a very good explanation as to what probably is happening, but I do think that the estrogen is doing something to the mitochondrial communication within the cell that's changing all of these things and causing our sex differences in not only the behavior, but also the molecular outputs that we're getting with my work and other work in the lab.
Great. Thank you so much, Gladys. And thank you to all of our amazing speakers from this session. Next, we're going to move on to our next session, and we'll have, or subset of speakers, we'll have 3 speakers. First up, we have Mariah Johnson with a pre-recorded talk.
Hello, my name is Maria Johnson. I'm a biology major at St. Joseph's University, and my presentation is entitled Dorsomorphin Can Prevent Memory Deficits Caused by Sleep Deprivation. Sleep deprivation is a common problem and occurs in 33% of adults, according to the CDC. Sleep deprivation can cause many issues, but the issue I'll be speaking about today is specifically the memory problems caused by sleep deprivation. The AMPK-mTOR pathway has been well implicated in memory consolidation. Sleep deprivation specifically affects memory consolidation. During normal sleep, mTOR forms a complex with raptor, which allows for protein translation to take place and memory consolidation to move forward. However, during sleep deprivation, levels of the regulatory molecule, AMPK, the activation levels are increased. This decreases the activation levels of mTOR. This then causes translation to decrease, which then means that memory consolidation cannot take place. Dorsomorphin is a selective AMPK inhibitor. This means that during sleep deprivation, it should attenuate levels of AMPK activation. This should allow mTOR activation levels to return to normal and protein translation and therefore memory consolidation to also return to normal. This is our experimental timeline for examining the effect of dorsomorphin on memory consolidation in mice. First, the experiment begins with a cannulation surgery. The cannula is placed to allow for direct injection of dorsomorphin or control saline into the mouse hippocampus, allowing dorsomorphin to bypass the blood-brain barrier. After 2 days of recovery, mice are then subjected to gentle handling for 4 days. This includes gentle handling for sleep deprivation as well as gentle handling for the object place recognition task. This is to allow mice to be acclimated to the experimental conditions prior to the actual experiment. The experiment begins with training for the object place recognition task, which I will go over in a further slide. Following training, mice are administered either dorsomorphin or control saline. This is done directly after training in order to make sure that we specifically impact memory consolidation. Following drug administration, mice are then subjected to 5 hours of sleep deprivation. The 5 hours of sleep deprivation are also done in order to impact memory consolidation in the critical periods which take place in 30 minutes and at 4 hours of sleep. Following this, mice are put back in their home environment until 24 hours after training, at which point testing for the object place recognition task takes place. The object place recognition task tests the mouse ability to distinguish novelty. The mice are trained for 3 sessions in an object configuration like the one shown in the figure below. After this, they are then allowed to have the drug administration, they are subjected to 5 hours of sleep deprivation, and then 24 hours later, one of the objects is moved into a novel location, making it a novel placement. This then is where the mice are tested. They're tested for 6 minutes to explore the novel object placement, and their preference for the novel object is measured in order to determine whether they experienced memory consolidation. Our current data suggests that sleep-deprived mice given dorsomorphin performed as well as non-sleep-deprived mice in the memory task. On the x-axis, the mouse conditions of vehicle mice given saline and dorsomorphin mice given dorsomorphin are shown. On the y-axis, their preference for the displaced object is shown. Specifically, we are examining whether the mouse explored the displaced object in a novel location for more than 33% of the total time it explored all of the objects. This would demonstrate that mouse memory consolidation did take place. We determined that the preference for the displaced object of the sleep-deprived mice was 47%. This demonstrates that the mice performed just as well and had just as much of a preference for the displaced object as both of the groups of the non-sleep-deprived mice, which means that memory consolidation did take place. This allows us to conclude that dorsomorphin can in fact prevent memory deficits caused by sleep deprivation. I would like to acknowledge my research mentor, Dr. Jennifer Tudor, Nicole Alicia, Natalie Burkert, Leah Gibbonus, Cameron Perot, Fatmata Sacco, Olivia Skarowski, and all of the other members of the Tudor Lab, as well as the organizations listed on the right. Thank you.
Thank you so much, Maria. Next up, we have a data blitz from Antoinette Perry. Oh, Antoinette, I think you're muted. There we go.
Thank you. Hi, my name is Antoinette Perry, and I am part of Dr. Ashley Ross's group, and I will be discussing the real-time detection of extracellular zinc signaling within the brain by use of fast-scan cyclic voltammetry. Next slide. So FSEV is a sub-second timescale technique that we use that actually has been used to study neurochemical signaling in the brain. Our most popular analyte is dopamine. So for this, we have our waveform where we have a holding potential of negative 0.4 volts, and we go up to our switching potential, which is 1.3 volts. And we scan over this waveform at 400 volts per second. Now, this is a lot faster than typical CV, which is on a millivolts time scale. So we are actually able to capture and study these redox reactions that are happening from these neurotransmitters. And that happens on a carbon fiber microelectrode. And this carbon fiber microelectrode is small, so where we can implant it in tissue, capture these redox reactions and not disturb the tissue and damage it. Next slide. So this is our modified Sawhurst that we use. And this is great because now we can co-detect purines as well as further stabilize detection of zinc. We capture the zinc reduction peak between the 0.5 and -1, and then we ramp up to 1.45 volts and hold for 3 milliseconds. And this gives time for the cleaning of the electrode surface, since zinc likes to deposit on the surface of a carbon fiber microelectrode. And the reason why we want to co-detect purines, that I will go into a little bit in the next slide, is because we would like to study further ischemia and zinc and purine correlation with that. Next slide. So co-detection of zinc and adenosine is possible with FSEV, and this is great. That way we can actually electrochemically see the interaction of zinc and adenosine that hasn't been seen when it comes to ischemic events using electrochemistry as a technique. So on our false color plot in the middle, we have our zinc reduction peak as well as our primary and secondary oxidation peaks for adenosine. Although we have that switching at 1.45 and we hold for 3 milliseconds, we actually can still detect the primary and secondary peak of adenosine where that is the actual oxidation potential that occurs at. Next slide. So here is our preliminary data that shows a promise of zinc detection by means of FSCV in vivo. So in panel A, we have circled our promised zinc transient peak. And then for B, we have our open CV. So that way you can clearly see this promised zinc transient peak denoted by the star. Now this larger peak is due to picospritzing that can occur when the electrode and the picospritzer is too close. And we need this picospritzer because we want to depolarize these neurons within the CA1, which is located in the hippocampus. And we want to be able to be there and see zinc because zinc is abundant within the hippocampus, more specifically the CA1, CA3, and the helicus itself. Now, zinc is electroactive, which is why we want to look at it because zinc is co-packaged and co-released with glutamate, which is an excitatory neuron within the brain. And this is actually something that we can look at since zinc is electroactive and unfortunately glutamate is not. So if we can quantitatively say that this is zinc and actually characterize this zinc transient or promised zinc transient, then this will be the first ever recording of zinc in the extracellular space by means of fast scan cyclic voltammetry as well as electrochemistry overall. Next slide. So in conclusion, this neurochemical detection of zinc is feasible using FSCV. Now we do have our promised zinc peak that was preliminary, and we have to further characterize to validate that this is an actual zinc transient. So for that, we will have to do pharmacological methods like blocking zinc receptors within the hippocampus and to make sure that what we're seeing is an actual zinc transient. With this now, we have further studying that needs to be done to optimize the mechanistic zinc release, as well as doing all this will actually lead to understanding zinc's role with ischemia, as well as the purines that are also linked to ischemia and other neurological conditions that zinc and glutamate are both correlated with. And that's it.
Thank you so much, Antoinette. Next up, we have Emily C. Kelly Castro.
Hi, everyone. Can you hear me?
Yes.
Yes, okay. So my name is Emily Kelly. I'm a PhD candidate at Rutgers University, and my thesis project is focused in understanding the role of a polarity protein called PAR1C on dendritic spines. And dendritic spines are those mushroom-like protrusions that you're going to see along the dendrites. And these structures are highly dynamic and highly compartmentalized. When I say that they're highly compartmentalized is if we do a close-up of a dendritic spine, we can see there are different levels of proteins like receptor proteins, scaffolding proteins, and cytoskeletal proteins. All of these proteins play an important role in shaping and in giving numbers to the different spines that would be expressed along the dendrites. Next slide, please. So why do we care about these proteins is because dendritic spines are really important for the formation of memory. So if any of these proteins fail, we're going to see changes in the plasticity of the dendritic spines and in cognitive functions such as learning and memory. One of the proteins that has been linked to neurological disorders such as bipolar disease and autism was this PAR1C protein. From previous studies in my lab, we were able to describe that this PAR1C protein regulates spine morphogenesis. So when this protein is knocked down, we're going to see that the density of the spines is decreased and the morphology of the dendritic spine changes. We also were able to determine that this PAR1C protein is activated downstream of the NMDA receptor. And once it's activated, it's going to phosphorylate the scaffolding protein PSD95 And when that phosphorylation occurs, that's what allows the dendritic spines to change in shape and size. So PAR1C overall, it's really important to regulate how dendritic spines are going to be changing when you're forming memories. Next slide. So one of our questions is, now that we know that PAR1C plays a role in spine morphogenesis, we want to know if we can reciprocate this in an in vivo model, and if we can see any changes in cognitive behavior. Therefore, we developed this Flux Mark1 mouse model where we breed the CAMK2 alpha Cre mouse line with the LOXP Mark1, and the CAMK2 Cre is only expressing the pyramidal neurons, what means that it's only going to be knocked down on the cortex and the hippocampal neurons. And here we see that we were able to validate that the PAR1 protein is being knocked down both on cortical and hippocampal neurons. Next slide. So one of the questions we were looking at is, how is the spine density and how is the spine morphology affected in this mouse model? So we were able to validate that spine density in our mouse model is being decreased compared to the data that we have in vitro, and spine length and width is also decreased compared to our control. And we're seeing that there's a more expression of stubby spines compared to mushroom spines. So what this means is that mushroom spines are the ones that are mature spines, while filipodia and stubby spines could be considered immature spines. Next slide, please. We also wanted to look at the cognitive defects, and we found that the Mark1 knockout mice exhibit an anti-anxiety-like behavior and are slower and have a slower working memory. So we did an elevator plus maze. What this means is if the mouse spend more time in the open arms, it means it doesn't have anxiety. And that's what we were seeing with our conditional knockout. Normally, mice are going to spend more time in the closed arms, which means that they have some level of anxiety. And we also were able to practice the Morris water maze, where we tested their learning memory. And what we saw is that overall, our conditional knockout mice are slower learners, but they still have good memories. And over here, I separated my data into males and females because there's a high difference between how males perform versus how the females perform. And that's it. Next slide. So I would like to thank my lab. I would like to thank everybody, all the undergrads that have helped me with this project, and the Neuro Mini Conference for giving me the chance of giving this talk. Thank you.
Thank you so much, Emily. Now we'll do a 5-minute Q&A for Maria, Antoinette, and for Emily. If you have just joined us, if you want to drop your questions directly into the Q&A box, and remember to indicate who they're for. First up, we have a question for Maria, or sorry, we have a comment for Maria. Someone said, great talk. We have another comment for Maria, someone also commending you on a great talk. And then they asked, I might have missed this, but did you deliver the drug into the brain globally or into a specific part of the brain? Why did you choose to perform why did you choose to perform the object recognition task? And do you think the drug is acting on the hippocampus? So a 3-part question.
Hello. So there are a couple of things to address the first part of your question. We actually we do surgery and insert the cannula specifically into the hippocampus bilaterally, so we know that we are targeting the hippocampus with the drug. For the second part of your question, the object place recognition task is known to be a hippocampus-dependent memory task, which is part of how we assess the effect of dorsomorphin on the hippocampus. So we have a pretty strong belief that the drug is in fact acting on the hippocampus because, again, of the cannulation surgery. And then, of course, with the deficits that we see in the mice that are sleep deprived would indicate that we are in fact impacting the hippocampus, since that is the part of the memory that would be impacted by sleep deprivation.
Thank you, Maria. Our next question is for Antoinette. They said, great talk and very exciting research, and asked, could you expand upon the contribution to zinc in ischemic stroke?
So the papers that I have read have linked zinc to reactive oxygen species, but not necessarily purines itself. And so a lot of work has been done on looking at adenosine and guanosine. So I want to be able to see if there is a correlation between zinc with this adenosine and guanosine that has been studied on FSCV without having to look at the reactive oxygen species. So there's correlation between what happens with elevated zinc in the brain after an ischemic event. And of course, if you have elevated zinc within the brain, you can go into neuronal injury, as well as if it becomes an issue, then excitotoxicity. So I want to be able to see what the correlation is and how that threshold looks of zinc after an ischemic event.
Thank you, Antoinette. We also had a follow-up question for you also about zinc. This person was curious if zinc acts differently in excitatory versus inhibitory neurons.
That I do not know because I'm more so wanting the excitatory. So there's a likelihood it will, but I'm actually just focused on what it does in an excitatory neuron.
Great, thank you. Our next question is for Maria. This person said they enjoyed your talk and they asked, is it possible to disentangle the possible effects of stress versus those of sleep deprivation?
Hello. Actually, well, we believe that any effects that we have for stress are, in fact, in all of the mice. So what all of the mice receive surgery, all of them receive an injection, and all of them who are sleep deprived, regardless of the drug or non-drug, are going through the same sleep deprivation process. So that includes tap shakes and disturbances to the environment. So any effects of stress should be seen in virtually all of the mice that are stressed.
Thank you, Maria. All right, if there are no more questions, we'll go ahead and move on to our next selected talk. And that is from Leane.
Yep, Leane.
Oh, Leane, sorry about that.
It's okay. Okay, so hello everyone. My name is Leane Ofaki, and the project I'm presenting today examines functional connectivity reductions in the default mode network of obese patients uncovered using MEG imaging. Next slide, next slide, next slide, next slide. Okay, so to clinically define the term, an individual is defined as being obese when their body mass index exceeds 30. Obesity is a highly prevalent phenomenon, contributing to over 60 chronic diseases, rendering it the leading cause of premature death around the world. Now, various factors contribute to obesity, including the environment through obesogenic diets, next slide, as well as genetic predisposition. However, given the neuroinflammatory response that results from obesity, which in turn gives rise to neurodegeneration, studies have shifted the focus to cognitive impairments that ensue obesity. Next slide. So this includes impairments to working memory, learning, and decision-making. Not coincidentally, all these functions are facilitated by an intrinsic brain network known as the default mode network, or the DMN. Next slide. Next slide again. So the DMN consists of frontal, temporal, and parietal regions, which exhibit high functional connectivity at resting state. So in other words, activity in these regions tends to be temporally synchronized. And previous studies have reported the implication of the DMN in internally directed tasks, such as introspection and self-referential thinking. Not only is DMN activity abnormal in obesity, as well as a range of other psychological disorders, but it's also been found to be anti-correlated with task-positive network activity. So together, these findings support the role of the DMN as a network that actively and deliberately engages or disengages during different tasks. So putting it all together, we hypothesized reduced functional connectivity in the DMN of obese individuals. Next slide. So as for the experimental methods, 24 controls and 6 obese adolescents were matched based on age and sex. Next slide. They were recruited to the SickKids Team Obesity Management Program Clinic at SickKids Hospital. Next slide. They were then subjected to the MEG scanner, where resting state network recordings were taken. So this was done with respect to 90 regions identified using the Automated Anatomical Labeling, or AAL, atlas. Additionally, because the MEG helmet records brain activity at a distance from the skull, additional statistical analyses had to be done to first eliminate noise in the data due to physiological artifacts, but also to differentiate the recorded activity to their respective emitting brain regions. So once done, next slide. Synchrony of pairwise cortical oscillations were identified with respect to 7 oscillatory frequencies, as shown here. Next slide. Next slide, yeah. So as for the results, the network-based statistics analysis toolbox, or NBS, was employed to identify significant network differences between the groups. So shown here are the functional connectivity links that were significantly reduced or impaired in the obese group as compared to the control group. And this was done with respect to the alpha frequency on the left and the beta frequency on the right. Note that the low gamma frequency also showed significant results, but the figures were not available for display. Nonetheless, I want to draw your attention to the regions indicated here in red, which pertain to the DMN. So, as expected, this includes frontal, parietal, and temporal regions, as well as the precuneus. So the NBS analysis toolbox has been useful because it enables identification of significance at the network level. However, more precise statistical corrections were then necessary to examine the significance of these functional connectivity patterns at the level of individual connections independent of network membership. Next slide. So to do that, false discovery rate, or FDR correction, was subsequently applied to the data. So similar to the previous slide, the functional connectivity links shown here are those that were significantly impaired in the obese group as compared to the control group. So as we can see here, for the alpha, the beta, and the low gamma frequencies, not only was activity within the DMN impaired, so DMN to DMN connections, but also connectivity between the DMN and external networks. So this includes the visual, the salience, and the sensory motor networks. I also want to draw your attention here to the posterior cingulate cortex, which I've circled in red, which was significantly affected in both the alpha and the beta frequency. Next slide. Lastly, the node degree analysis was computed to identify affected brain regions with the highest degree of within and across network interactions. And what we found was that regions pertaining to the DMN were among those highly implicated. But more significantly, I want to draw your attention to the fact that in both the alpha and the beta frequency, as shown here, the posterior cingulate cortex was the region with the highest node degree. Next slide. And the significance of that will be discussed shortly. Next slide. So in summary, we found that obesity resulted in functional connectivity impairments, which spanned networks. So impairments within the DMN, as well as between the DMN and external networks, such as the visual, the salience, and the sensory motor network. We also observed these significantly impaired functional connectivity patterns in the alpha, the beta, and the gamma bands. This aligns with findings of previous studies implicating these oscillatory frequencies with internally directed tasks. Lastly, the FDR analyses depicting the intra and inter-network impairments also supports the view of the DMN as a hub of transient functional interactions. So, this hub is connected to various other brain regions and thus serves to integrate information of different modalities to enable scene construction. In turn, this enables the execution of those internally directed tasks. And a key player in this hub is the posterior cingulate cortex, which also plays a key role in this integrative function, as observed through the node degree analyses, the FDR analyses, as well as the findings of previous studies. Next slide. Next slide. Now, the significance of this research emerges when we consider the grave health risks that obesity contributes to, in addition to the lack of integrative research in this topic. Next slide. If we identify a brain region, a brain network that's underactive as a result of obesity and is also implicated with the symptoms observed, then perhaps stimulation of this brain network can help ameliorate the symptoms and thus reduce the prevalence of obesity. So this could be done through the brain stimulation techniques such as transcranial magnetic stimulation, TMS, or deep brain stimulation. Now, one limitation of the study is the fact that many factors contribute or mediate the relationship between obesity, um, mediate the development of obesity. So, as mentioned earlier, this includes diet, lifestyles that lack movement, genetics, as well as high levels of circulating triglycerides. Future studies would benefit from teasing apart the influence of these contributing factors through a controlled paradigm. So, for example, one sample would exclusively report poor eating habits, another would report a sedentary lifestyle without being obesogenic, and thus the functional connectivity of these samples would be compared to an obesogenic cohort, such as the one employed in this study. Additionally, given the correlational nature of the relationship between the DMN activity and the symptomology observed, we can't infer any causality. So to overcome this, the brain imaging data that we obtained here could be supplemented with behavioral data to further support the findings of this study. Lastly, I think it would be interesting to look at whether the extent of DMN underconnectivity actually correlated with behavioral measures. So that could include measures of intelligence, introspection, and any other cognitive function that we know are associated with the DMN activity. Next slide. So these are the references. Next slide. Thank you for listening. Any questions?
Now we'll take questions for Leane. So if you have them, if you just want to drop them in the chat. We have one from Maria. They said, great talk, and asked, did you see any differences in sex or age given that participants were age and sex matched?
So we haven't done that analysis yet. I think another confounding factor would be, so our obesogenic cohort was only 6, an N of 6, so very small sample to conclude any differences yet. But I think that would be interesting to look at for future research.
Thank you. Someone else said, very nice. I imagine that there's comorbidity between obesity and psychopathology. Mm-hmm. And they asked, do your results suggest any association with either anxiety or depression?
Mm-hmm. That's a great question. So we know, as I mentioned early in the talk, we know DMN abnormal activities associated with other psychological disorders, but I haven't exclusively looked at obesity and psychological disorders. But then I think given the complexity of both the DMN abnormal activity and then the way it manifests in obesity, but also the way that the underconnectivity could be associated with other psychological disorders, I think it would be an interesting area to look at as well.
Thank you. We have another question. They said, thank you for your talk, and asked, do you see any networks that showed overconnectivity in the obesogenic cohort?
So another good question. I did not see, so the data I presented was all hypoconnectivity, so reduced connectivity in the DMN, and that's what I focused my study on. But I don't think there were any networks that showed hyperconnectivity.
Thank you. Another comment, fantastic talk. And
thank you.
It's known that obesity increases the risk for the development of mild cognitive impairment and Alzheimer's. To what extent do the obesity-linked DMN impairments that you've characterized recapitulate the functional deficits seen with Alzheimer's disease, such as posterior cingulate cortex deficits, etc.?
Okay, give me 1 second. Let me just reread the question.
Yeah, it was a dense one.
Mm-hmm. So if I understand the question correctly, they're asking if, oh, they sent a follow-up question.
They just said sorry for it being
totally
complex.
fine. if I understand the question correctly, they're asking if there's any similarities between the deficits that I observed in the obese group, if those could overlap with, if those would overlap with Alzheimer's disease. Am I interpreting the question correctly?
I think so.
I think once again, I think it's worth mentioning that this area of research is still fairly new. Even when doing my own background research, there wasn't much to go about. Much of the available research was mostly with looking into eating habits, not even obesity in itself. So I think, again, that would also be an interesting area to look at. Yeah. Given, I think also, again, given the interactions or similarities between the brain connectivity deficits that we see both in obesity as well as these other psychological disorders.
Thank you. We'll have one last question for you. This person also said, great talk. And they asked, in your network analysis, how similar are the network's hub structure across frequency bands? Is the PCC the only node consistent across frequency bands?
That's a good question. So I don't think I kind of assumed that people would know much about the frequency bands. So the only significant bands that we cared about were the alpha and the beta and the low gamma to an extent, because those three frequencies would be the ones that are expected to be involved in cognitive tasks, especially if you're at rest. But to answer your question, yes, the posterior cingulate cortex was the one to, it was the only node to show consistent, to be both highly implicated as well as to be affected. I hope that answered the question.
Yeah, thank you so much. Thank you for an amazing talk, and thank you to the audience for those questions. Next, we're going to go ahead and move on to our next 4 talks. Thank you again.
Thank you.
All right, next up, we have a talk from Dwaynica Greaves.
Hello. Hello, everyone. So my name is Dwaynica Greaves, and this research is going to be focusing on the effects of performing arts on social cognition and engagement. I am a PhD placeholder at University College London, and I'm studying, should be studying cognitive neuroscience. And this talk is going to be focusing on specifically the repurposing of health technologies for research in performing arts. So next slide, please. So yeah, the division of neuroscience that I'm in is neuroaesthetics. And this is a novel field of research which fuses fields such as biological theories, psychology, neuroscience, to investigate the socio-cognitive processes that contribute to the creation and the aesthetic response to art. And if you press next, you'll be able to see where I lie in all of this. And my research focuses on performing arts in dance and theatre, but today I will be focusing more on theatre and also psychology. My undergrad is in psychology, so I'm interested in looking at the social engagement and cognition. And neuroscience comes in for me, which is quite new as well, because I've only started this kind of area of research, is to look at the neural correlates of the above. So next slide, please. So I'm going to be talking to you today about my project that I did for my master's, and it was a massive project, collaboration between Goldsmiths and University College London, and we were looking at actors and the audience as well. So this section focuses on the actors. So if you press next, you will see that our background was basically, our name is a powerful social signal, and in infants and adults, hearing your name engages the medial prefrontal cortex. But can this be affected by context? And by context, we basically meant acting versus non-acting. If actors respond to their name, what does it elicit in the brain if you're in character and when you're outside of character? And to do this, if you press next, you'll see that we use FNIRS, and this is in the simplest way, the portable fMRI, and it uses infrared light to measure blood flow in the surface of the cortex. And if you press next, you'll see that we found there was strong activation in the prefrontal cortex when you heard your own name when you were not acting, but there was actually stronger activation when you heard your own name when you were not acting. And maybe this could be because when you are acting, you're maybe using more higher functions, you're mentalizing a bit more. And maybe it could be because when you hear your name, you maybe snap out of your character and you're grounding in yourself. So if you go to the next slide, you'll see what I wrote my final project on was actually the audience side of it. And I was looking at the effects of audience participation on audience engagement in these theatrical performances as well. So if you press next, you'll see that my rationale was about breaking the fourth wall. What happens when you take the audience member and put them on the stage? And going into the neuroscience a bit more, in terms of mirror neurons, you know, when you see someone do something, it elicits the same neural processes in the brain as if you're doing it yourself. But the question is, if you see it and now you're actually doing the action that you're seeing, will that make a difference? So we designed a collaborative performance. So it had a scientist explanation explaining the findings from the previous study. It also had a participatory period, which is what I used to conduct my experiment. And it also had the actors that were acting out a deconstructed version of Midsummer's Night Dream, hence the name of, you know, the whole project. So I was looking at audience participants versus spectators, and the participants went on the stage and the spectators were the ones that sat on their seat. So if you press next, you'll see that I use the Empatica E4 wrist sensors, and this company focuses on health tech, and this specific device is used for researchers to collect all this data. As you can see, the data that it collects, but I focused on heart rate. And I also collected self-report measures as well, because for me, I guess coming from that psychology background is really important for me to see what the audience members are thinking about as well. So if you press next, you'll see that, well, we didn't actually find an effect of participation on heart rate, but we did find effect in the self-report measures. We found that participants actually felt more bonded to the scientist performance, because I said before, there were scientists on the stage, there were actors on the stage as well. So they felt more bonded with the scientist performance. And I kind of think that this is because the whole performance was for the purpose of science, but I'm not going to go into it because it's a lot, but it's the fact that maybe context and knowing why you're there, maybe that has an effect on how you feel in terms of engagement with the people that are on the stage. So next slide, please. Yes, so where am I going with this? So for me, what I hope to do for my PhD is basically look at the limitations of the previous study and think about how I can replicate this, how I can improve this. So for starters, the participatory period was actually quite short. It was about 10 minutes. For me, I want to make this longer. I want to look at forum theatre, which is a style of theatre where you are immersed in the performance, you control the narrative, you're solving social issues, and perhaps that might elicit more processes than just watching a performance and maybe having a little participatory period. Second of all, heart rate may not be the strongest measure. So I want to look at acceleration, which is collected on the same devices. I look at unconscious mutual synchrony, which the researchers that I was working with, they used that before. And through that, you can actually learn more about engagement as well. And I also want to improve my self-report questions and make sure they're more focused on engagement. And the implications of all of this, so why this all matters, is because in especially the UK, we can see that the arts doesn't have such a high status compared to other subjects, as we can see the struggles that the UK is going through right now in the arts. And for me, it's really important to make sure that research is interdisciplinary, that we can see how we can pull things from the arts, from neuroscience, and conduct research to better the educational resources that we have. And lastly, to validate the repurposing of health tech and seeing how all these devices that we use, how can we use them for different sections of neuroscience or different sections of psychology, and conduct our research using these multiple devices. So thank you so much for listening to my talk. Thank you, Black in Neuro, for having me here today. If you want more information, please contact me as well. And I will send a link to the chat with the website of the project, and all the contributors are basically there. So thank you very much.
Thank you so much, Doinica. That was great. Before we move on, I want to take a moment to introduce our judges for this section. We have Dr. Thorne, who is Catherine Thorne, who is an assistant professor at the University of Texas at Dallas. Her lab studies neuroplasticity and the motor system. We also have Sama Ahmed, who is, a presidential postdoc fellow in the Murthy Lab at the Princeton Neuroscience Institute. And last up, we have Dr. Rahila Tarfa, who has a PhD in neuroscience and is now in medical school studying therapeutics for tinnitus during a year off from their research fellowship. So thank you so much to those judges for being here. And thank you to our judges from the earlier session. Next up, we have Ben Baker. Thank you so much.
Hey, thanks a lot for having me, Black and Neuro. So I'm a postdoc right now in Conrad Cording's lab in neuroscience. So I'm kind of an aspiring neuroscience. This is a little bit new. My PhD is actually in philosophy. And so I hope you'll pardon the bumping up against disciplinary boundaries and change of pace. This is really going to be a philosophy of neuroscience paper that right now I'm working on with Conrad and Ben Lansdell. And so those are my PhD supervisors in philosophy who influenced my thinking on this a lot. So this is going to be kind of a big picture philosophy thing in 5 minutes. So here we go. Next slide. So here's the basic issue that we're concerned with. Thinking about semantics around the terms information, these italicized terms, information, code, computation, and representation. So a lot of these terms are used to interpret the results of experiments that essentially probe correlations between neural activity and controlled variations of stimuli or behavior. So a lot of the talks that we've just heard from actually weren't talking about neural representations, and Dwanica's talk just now had used more careful language around neural correlates. So that was nice to see for me. From my perspective, I'm interested in the term representation and how it's used. And here I want to kind of hide behind a couple neuroscientists, so it's not just my philosophical opinion on this, that these terms are used in kind of an unsystematic way. So Conrad Cording has a paper on this topic in 2017, and more recently Brett talks about the neural coding metaphor in critical terms that I'm building on. So next slide. So here's big picture how philosophers have thought about representation. Defined to involve two sorts of things. One is content, that is, representations are about something or they're directed at something. You might just say they mean something. And representations come in different types or modes, so you can have the same content represented in different ways, basically. These features are often implied in the use of as and of. So here are some examples. A map of pen is a representation of pen. Pen would be the content. And then here's my gloss on the mode of representation of a map, something to be navigated. A painting of pen, also content pen, different mode, say something with a certain look. An utterance pen, again, representing pen as, let's say, something to be recognized by the listener. So the general approach in philosophy of trying to account for mental stuff, beliefs, desires, intentions, etc., in terms of representations is known as representationalism broadly, and this has a long history in philosophy. The sort of explanatory purchase you're supposed to get here is that representations of various kinds can be false, they can have errors, they can be inaccurate, like a map can be inaccurate, they can not cohere well together. And these are things that we think mental states do. They can be inaccurate, you can exhibit flaws in reasoning. And so representationalism is supposed to explain the possibility of error, or it's supposed to be a route to explaining the possibility of error. The puzzle you come up against when you go down this approach is, okay, what actually determines the content of the representation? What makes the difference between the error cases and the success cases? Next slide. So just some, I think we might have missed a slide. So just, yeah, to quickly mention some historical figures here. We've got representationalism showing up in ancient Greece, in Aristotle's account, that has kind of both form and matter. So it's dualistic in the nature of substances. In Samkhya Yoga, you have a physicalistic view of representations where in the mind there are these vrittis. The mind has these different components. The vrittis are physical changes that refer to the outside world. In the medieval period, you have some very influential Arabic philosophers talking in terms of mana, which then later is translated into intentio. And in the West, you have intentional theories of mind found in Descartes, Locke, Hobbes, and others. All of these are, very different metaphysical pictures, but all relying on the notion of proper states of the mind that represent states of the world, etc. Next slide. So the main notion of function that we want to think about in this paper is a teleological notion of function. So, sorry, let me pause for 1 second. So representationalist theories today rely on some normative notion of function. And we're going to talk about the teleological notion of function, which is a prominent view in philosophy of how this works. The thought is when you're thinking about neural representations broadly, you're not just thinking about information. So if you look at my smaller bullet points here, the heart is something that we think of as having a proper function to, say, circulate blood. If you're thinking of the neural activity of N, not just as carrying information, but as having a proper function to carry certain information and not other information, that's the way to build up towards your theory of representation. And teleological views of function are the view of function that we talk about in this paper. Okay, next slide. Okay, so, all right. So there's basically two kinds of teleological views of function. You can look backwards at what a part of a system was selected for, or you can look forward at how it contributes to a particular goal. On either approach, you're going to have a framework for thinking more specifically about neural representation than kind of what's on offer today is the idea. And so what we're trying to do in this paper is look at various approaches to C. elegans research that draws on kind of whole brain recording, and use this teleological, sometimes called teleosemantic approach in philosophy to chart out some different hypotheses that you might make about what's being represented in different parts of the neural system, not just on the basis of what information is there, but on the basis of specific claims about function, either on this backwards looking or this forward looking approach. And so that's it. Thanks for being with the philosophical stuff, and thanks Black and Neuro for having me, and thanks for listening, everyone.
Thank you so much, Ben. Next up, we'll have a talk from Yasmin Lowe.
Hello. So my name is Yasmin, and I'm a senior in Dr. Margaret Bell's lab at DePaul University, and we study the my talk will be about the perinatal exposure to environmental contaminants, or PCBs, altering hedonic behavior during adolescence. Next slide, please. And so what PCBs are essentially, PCBs are polychlorinated biphenyls, and they're an environmental contaminant that were commonly used in paints and industrial materials before they were banned in the 70s. And because of their long half-lives, enabled them to remain in the environment long after their use is stopped. And PCB exposure can occur early in development via maternal exposure or transmission in utero or through lactation. And so why do we care about PCBs now, essentially? And so PCB research has shown that PCBs are implicated in affecting the dopaminergic and estrogen signaling, and our lab has recently found that they also have an effect on microglia. And these endpoints are vital in reward seeking, social, and anxiety or depressive-like behavior. And so what we hope to determine through this experiment is the effect of perinatal exposure of PCBs to affective behavior and ethanol intake in male and female rats. Next slide, please. And so to do so, we ran a series of behavioral tests, and one of which I'll be talking to you today, which is the bottle preference test. And so what we did is that we orally exposed Sprague Dawley dams to an environmentally relevant mix of PCB or an oil control daily during pregnancy. And then we also monitored the physiological development, such as body weight, age of pubertal onset for the male and female pups. And then at adolescence, male and female rats were singly housed, and they underwent a bottle preference test over the course of 11 days. And this was served as a measure of hedonic or reward-seeking behavior. And then half of them were tested for a choice between water or an ethanol solution, in which the concentration ranged from a 3% to a 12%. And the concentration gradually increased, and it was also sweetened with saccharin over time. And then the second group was also had a choice between water or a saccharin solution, in which the concentration was either 0.25 in the beginning or 0.75 at the end. And then the bottles were weighed and refilled daily to determine the intake of each liquid. Next slide, please. And so what we found essentially was that PCBs, they increase the sweet preference. So as you can see in figure 1A on the Y, it's plotted with a volume of saccharin intake over the total volume intake. So this is water and saccharin together. And that we found that PCBs prolonged high saccharin intake relative to controls, and the male and females combined, as statistically we didn't see any main effect of sex. And so you have the top, you have the black line in the figure 1A, which is the PCBs, which is significantly greater than the pink line, which is the oil. And this is across days. So it's the 0.25% solution as well as compared to the 0.75 solution. And then in to the right, we have figures 1B and 1C, and this is plotted with the the massive saccharin relative to the body weight. And although we didn't see any sex effects, this is just plotted to sex just to demonstrate that. And so we saw that PCBs increased the saccharin intake at higher concentrations. So at that 0.75% solution, we saw that PCBs increased saccharin intake. And this is, although there wasn't any sex effects, this was more pronounced in females, as you can see in figure 1C. Next slide, please. And so we also found that PCBs induce only minor changes in the voluntary ethanol intake in males. And so as you can see in figure 2A, it's volume of ethanol intake over the total volume intake, so similarly to the saccharin graph as well. And so there was a significant effect of day as intake increased with a greater concentration of ethanol, but there wasn't any main effect of sex or PCB exposure, as you can see in figure 2A. Although an interaction was present between sex, PCB, and day, as you can see on day 5, that star, but the source couldn't be determined statistically, although it does appear to be, it could be an effect because of the male PCB group. So this is seen in figure 2B, in which there wasn't any main effects of PCBs, there wasn't any main effects of PCBs, but you can see that this is figure 2B and 2C. It's plotted what the ethanol consumed relative to the body weight. And so males exposed to PCBs did have a greater ethanol intake at the 0.6% concentration, but this is not seen in other concentrations, such as the 12% concentration for figure 2C. Next slide, please. And so overall, we found that PCBs appear to potentiate sensitivity to sweet reward in both males and females. However, they don't significantly alter the voluntary of ethanol intake. And this could be because of the known effects of PCBs in potentiating dopamine release in the mesolimbic pathways. And as previously stated, we ran a series of behavioral tests, and although the analyses of those are still ongoing, but the behavioral tests that we'll run are the forced swim test, which is a measure of depressive or anxiety-like symptoms, play behavior, which is a measure of social reward, a light dark maze, which is the measure of anxiety-like behavior. And then we also, 1 hour prior to euthanasia, rats received a bolus of either water or ethanol, and then their brain tissues were collected, in which we'll run qPCR or immunohistochemistry to study the mesocortical limbic system, and will determine the effects of PCB exposure on microglia and dopaminergic responses in response to that ethanol challenge. Thank you so much, Black and Neuro, for having me, and I'd like to just give thanks to DePaul University and my mentor, Dr. Margaret Bell, and my lab mates. Thank you.
Thank you so much, Yasmin. Our last data blitz before our next Q&A will be from Alyssa Maycumber.
Hi, everyone. Thank you so much for having me. My name is Alyssa, and I'm going to be talking about the effects of arterial stiffness on verbal learning in non-demented older adults, over from UC San Diego under Dr. Mark Bondi and Dr. Granholm. And next slide, please. Okay, so the relationship between vascular risk and cognition has been well established, and there is substantial evidence for a link between vascular burden and the development of dementia. And so our elevated arterial stiffness, which is increased systolic blood pressure and pulse pressure, is a result of structural and functional changes of the vessel wall that occurs with aging. And so pulse pressure is looked at to be a risk factor for the development of cognitive and functional impairments. And verbal learning has been observed as a reliable neuropsych marker of cognitive decline in early AD, along with preclinical Alzheimer's disease. And there has been limited research to evaluate the association between arterial stiffness and verbal learning in participants, specifically with mild cognitive impairment versus those healthy controls. Next slide, please. And so our study looked at 66 older adults, 53 were diagnosed as cognitively normal, and we had 13 MCI. And their blood pressure was taken at the assessment with us. And pulse pressure was used to estimate arterial stiffness. And the formula, which is the last bullet point at the bottom, removes the effects of systolic blood pressure. And we did also look at systolic and diastolic blood pressure independently. And all blood pressure variables were log transformed to improve normality. And so for verbal learning, we use the CBLT2 trials 1 through 5, which is when we read a list of words over a course of 5 trials, and they repeat it back to us after every trial. And so we used a multiple linear regression, which evaluated whether cognitive diagnosis moderated the association between arterial stiffness and verbal learning. And we controlled for age, sex, education, and diabetes status. Next slide, please. So our results, the moderating effect of diagnosis on arterial stiffness and verbal learning, there was significance. And so when we looked a little bit closer at just cognitively normal individuals between arterial stiffness and verbal learning, there was no significance, which you can see to the graph, the blue purplish color. But when we did look at just the MCI individuals between arterial stiffness and verbal learning, there was significance, which is the red portion of the graph, which shows that increasing pulse pressure in mild cognitive impairment individuals was associated with worse learning. Next slide, please. And so our conclusions, arterial stiffness highlights the utility of this as a marker of changes in cognition in older adults with mild cognitive impairment compared to those traditional blood pressure measures. So clinically, arterial stiffness could be used as a non-invasive and cost-effective biomarker for individuals who have been diagnosed with MCI or who are at risk for Alzheimer's disease. And some future directions is maybe examining physical activity as a component of this, along with maybe throwing in cognitive reserve or other neuropsych measures. And I would personally like to increase the MCI sample. And I think it would also be interesting to look at Alzheimer's disease individuals as well. And of course, adding diversity in participant populations, since we do know that there are disparities within cardiovascular and neurodegenerative conditions. Next slide, please. So this is my contact information. Feel free to reach out with any questions or if you just want to reach out to chat. Thank you.
Thank you so much, Alyssa. Now we'll take questions from all 4 of our speakers from this session. We'll take questions for Doinica, Ben, Yasmine, and Alyssa. So if you want to just go ahead and drop them in the chat, I'll ask them live. Our first question is for Doinica. This person said, really neat. People have done something kind of similar, but with musicians performing and an audience, and have looked at measures of EEG synchrony among audience members and between audience and performers. Have you done anything comparable?
Okay, so that's actually quite interesting because I've actually been in recent talks about how EEG might actually be better to use than FNERS. But for me personally, I started this research last year and I hope to continue it, funding permitted, next year. So I'm in a place where I'm actually still reevaluating what device would probably be the best to use as well. So it's not something that I have done, but something that I definitely want to look into.
Thank you. There's another question for you. What's the importance of taking arts research out of the lab? Why did you decide to make this methodological choice?
Okay, so I've got 2 little answers for that. So the first one, my first study I actually conducted was in the lab, and it was on the effects of mood induction on emotional perception of movement stimuli. And I was looking at ballet dancers, etc. And I found that I feel like the lab research didn't encompass what I want in terms of the ecological validity of being in a theatrical space. I feel like I would use lab research if it was well suited to the question. It's all about what is best for the hypothesis. And the second thing is that I'm really passionate about having interdisciplinary research. I am an interdisciplinary person, even outside of my research. And for me to be in the theatrical space, I mean, that's where I get my energy from. I feel like it makes me a better researcher when I am in a place where I can be as authentic as possible, because that makes my research as authentic as possible as well. So thank you for those questions.
Thank you. This next one is for Dr. Baker. The comment at first was really important work and important for keeping experimentalists like myself from getting sloppy or lazy. In cognitive neuroscience, there's a lot of emphasis on multivariate decoding and encoding methods. If one can decode from brain activity whether someone is, say, looking at pink versus red, can we call that a neural representation?
Great, yeah, thanks for the question. So on the sort of teleological approach we're pushing in this paper, the answer would sort of be it depends. So you might think you're looking at neurons in V1 or something, and have a more or less explicit hypothesis that these neurons function to what they're supposed to do is carry information about color, because we know upstream from that there's something else having to do with visual processing. By contrast, if you were able to decode from PFC, you might worry that something weird is going on. Maybe this person just had some traumatic experience with red, and that's why we actually have this information in this part of the brain that we don't, a priori, I think, has that sort of function. So it's kind of, the thought is you need to go beyond code, but informed by hypotheses about function, decoding methods could be quite useful.
Thank you. This is for Yasmine. This person said, super interesting talk, and they asked, have you examined whether rats exposed to PCBs exhibited any changes in consumption of rewarding solids versus liquids?
Hi, thank you for your question. We actually haven't considered looking at it from a solids versus liquids perspective, but I think that's actually really interesting that you bring up that point, just because of the consistency textures as well as preference in terms of liquids. We put it into a cookie wafer, so it was a lot, so they ingested it that way. But I think it's really interesting just for it to be a solids versus liquid aspect, and I think that's actually a really interesting take to look look at it, and I think that we should actually really consider that. Yep. Thank you.
Thank you, Yasmine. This question is for Alyssa. The comment was very nice, and they asked, would including a measure of verbal fluency add sensitivity to your behavioral measures?
Yeah, so it's actually something that I am also interested in verbal fluency, and our lab does a comprehensive neuropsych battery that includes that. And so I think it would be interesting to see if there are any correlations between verbal fluency and verbal learning, and what that would look like across different diverse populations. Thank you.
Thank you so much. All right, thank you for those great questions from the audience, and thank you to all of our speakers in that section. We are at our last section of speakers, and so we'll have 5 data blitzes. First up, we have Jamal Williams.
Hello, everyone. My name is Jamal Williams, and I'm a graduate student at Princeton. And today I'll be talking about how higher order brain areas represent the high level event structure in music. So to motivate this, in trying to understand how we segment rich continuous stimuli into meaningful chunks, researchers have discovered a set of brain regions which represent naturalistic stimuli at varying timescales. The findings above, for example, show that as fMRI participants are perceiving a TV show, higher order brain areas, such as precuneus, highlighted in orange, inherit the short timescale information from sensory areas, in effect abstracting away from low-level features to form representations of high-level information, such as important scene changes within the episode. So this led me to ask whether these same higher order areas represent the high-level event structure in music. To address this, I designed an fMRI study where I had 25 participants listen to 16 songs, 8 of which were classical and 8 that were jazz. And then we had a separate group of behavioral participants listen to each of the songs and were told to press a space bar anytime they felt a meaningful change occurred within the song. From this, we were able to derive a set of event boundaries for each individual song. So to identify which brain areas chunk music in the same way as our human annotators, we use a searchlight approach whereby we repeatedly take the song-specific time courses from a subset of voxels in the brain, and then we fit a hidden Markov model, also known as a HMM, to this time series. To briefly elaborate on the fitting procedure, what the HMM is attempting to do is it's attempting to represent the fMRI time series as a set of stable periods punctuated by shifts between these periods. After fitting our model, we then compare the boundaries from the model to the boundaries provided by our human annotators, here in red. We counted the pair as a match if the human annotations fell within 3 seconds of an HMM boundary. So we would say for this toy example that there was a match here, here, and here. We then quantified this match by comparing the true match score to match scores from a null distribution of shuffled boundaries. The brain areas which had the best matches to the human annotations across all 16 songs are shown here. Based on previous research, we predicted that auditory cortex represents low-level musical information, and higher order areas such as angular gyrus and precuneus contain abstracted representations of the music which don't depend on the low-level musical features explicitly. To test this prediction, we asked which brain areas would preserve its event structure even when we remove the low-level musical features from the fMRI signal and then refit the HMM. Our primary hypothesis being that matches in auditory cortex would be most negatively affected by this feature removal, whereas higher order areas would be least affected. And this is what we found. In the plot above, starting on the far right, matches between the HMM and the human annotations get worse in auditory cortex after the feature removal, while precuneus and angular gyrus are less affected by this. Furthermore, precuneus is most resilient to this feature removal and practically maintains its event structure relative to the other ROIs. Finally, if we take a closer look at the types of representations that exist in each ROI, we see again, starting on the far right, that auditory cortex is representing the short timescale information in the music, whose event boundaries do align with human annotations, but are lost when we remove the low-level musical features from the fMRI signal, as shown on the previous slide. As we march towards the left, we see the representation become more and more abstract as we make it to precuneus on the far left. So to conclude, I highlighted 3 brain regions that chunk music similarly to our human annotators. Specifically, that was A1, angular gyrus, and precuneus. Precuneus and angular gyrus are a part of the brain's default mode network, which has been shown to represent high-level narrative information, and these regions appear to have abstracted representations of music and are relatively impervious to the removal of low-level features. Finally, auditory cortex also represents musical information, but in this case, it was mostly low-level features. So with that in mind, I would just like to say thanks for listening. Thanks to my labs below at Princeton, and I want to also thank the NIMH and the DSPAN for funding this research. I'd be more than happy to take any questions if anyone has any. Thanks.
Thank you so much, Jamal. Next up, we'll have Freedom Chiteni.
Hello. Hello, everyone. My name is Freedom, and I'm over here with the Stanford Center for Compassion and Altruism Research. Next, I work with Dr. Doty and Dr. Carl Deseroth within the Department of Neurosurgery within the Center for Compassion and Altruism Research. Next. So, well, we are a very, very unique lab in that we are looking at neuroscience and vascular brain surgery and imaging all together from the perspective of human behavior itself. So while science has made like great strides in treating pathologies of the human mind, far less research actually exists to date on positive qualities of the human mind, including compassion, altruism, and research. And how do you actually measure that? Yet these positive pro-social traits are so innate to us and lie at the very, very centerpiece of our own common humanity. And especially today, if you look at what's going on in the world, we don't have to go too far away to look at the neurological basis of compassion for humanity. So our capacity as humans to feel compassion has really ensured the survival and thriving of our species over millennia. And that's what our research is really grounded on. And then for this reason, what our work at the Center for Compassion, or CCE, has the explicit goal of promoting, supporting, and conducting scientific research studies of compassion and altruistic behavior. So next. So next. So founded really by my advisor, my mentor, Dr. Jim Doty, who is a clinical professor of neurosurgery and my PI at CK within the Department of Neurosurgery. We really work on on collaborations with a number of prominent neuroscientists, behavior scientists, and geneticists, and biomedical researchers to closely examine the physiological and psychological correlates of compassion and altruism. And the mission of our lab really is to investigate the methods of developing compassion and promoting altruism within individuals and society through our research. So you'll find that my presentation today is really going to focus maybe on one aspect, but our lab really looks at a grand scheme of things, around the neural correlates and the biological basis and probably the antecedents of compassion. So next. So I'm going to look at optogenetics. In fact, that's the one thing I'm going to focus on today. And this is something that was discovered by one of my PIs, Dr. Daiseroth. Next. Next. Yeah. So what optogenetics is really founded by Dr. Cole himself, and in collaboration with Dr. Doty and myself, it's a new field that actually came out of our lab. And the goal for this really was to really think of new ways to look at the enormous efforts of clinicians and researchers to expand their insights into psychiatric disease, and to figure out new and interesting ways and techniques really to figure out what's going on in the complex mammalian brain. So what we're proposing here is to use this optogenetics technology to study the mammalian brain from the perspective of compassion itself. So next. So the goal really was to use optogenetics to study the neural basis of social compassion in the mammalian brain. So, and our team is undertaking a focused and ambitious definition of the causal neural codes that underline the most fundamental of human social behaviors. Next. And our methods here is that we're actually using mutant mice lines with altered social behavior, including stunted nurturing, decreased sociability, and increased social behavior. So nurturing is a fundamental form of socially compassionate and empathetic behavior that can be readily studied in animals. So the mutant mice that we use with this altered nurturing leave their young scattered around, for the next, to die of exposure. So what our team is doing with the Daiseroth lab is to really look at this optogenetic process as a more effective way of controlling a neuron's activity using light and genetic engineering. And so we are essentially focusing on this protein called opsins, and opsins occur naturally, and they were first discovered in algae. So the technique that we have created and the tools that we are creating look at the mouse to get the opsin into the neurons of the mouse. And the genetic code for the opsin must be carefully inserted into the genetic code for the neurons in that mouse. And so if this is done correctly, every neuron in that mouse should have the opsin, and that would alter its behavior. So because now we understand a lot about the genetic code of the mouse, we can choose where to put the opsin. We can insert the code into a specific type of neuron or into a specific location in the brain, and we can choose exactly which neurons we want to control. And the most popular opsin that we've been focusing on is the CHR2, or the channel rhodopsin 2. And again, if you are in that field, you would know that it comes from the green algae. So this protein is actually activated by blue light, which means that it only works when blue light shines on it, and it doesn't respond to any types of light. So when we have this protein CHR2 into the neurons, it means that the neuron can be turned on with blue light, and it can be on for as long as the blue light is shining on them. And so what this does is it gives us precise control over the timing of the neuron's activity. So normally neurons are not affected by blue light. So only the neurons that actually have the CHR2 will be affected by the blue light. So next. So what we are predicting is that when we use our optogenetic tool to see if these mice can switch on the social behavior associated neural networks in mice exhibiting a lack of compassion to their young and change them into parents exhibiting socially appropriate care. In the others, I think we're also predicting that we will be able to analyze, their social approach, social novelty, and social communication. And I think the benefit of our work really is fundamental in that we are looking at how our observations in this research could really be applied in creating a world that is more compassionate in a way that, especially today, where we are seeing a lot of turmoil in the world. And so we have a movement right now called the Compassion Revolution in our lab, where we're looking at what is the role of neuroscience in terms of impacting how everything works. Next. So we have built a technology. There's an app, you can find it in the iOS, called Alphabet of the Heart, and that's always evolving, and we are always looking for anyone who is interested in social compassion, and also being able to look at how this optical deconstruction of altruistic behavior from the lab can translate into the grander scheme on how humans behave. And I will just leave this because of the short time period that we have, but I'm happy to answer any questions, or if you guys want to learn more about our work, you can email me. I didn't put my email here, but it's freedom@inventxr.com.
Great, thank you so much, Freedom. And we'll get to your questions after the next 3 talks. Thank you so much.
Welcome.
All right, next up, we have a pre-recorded talk from Samaya Dyer.
Hello everyone, my name is Samaya Dyer, and today I will be talking about a study entitled Adolescent Predatory Stress Increases Peripheral TNF-alpha Response to LPS Regardless of Sex. So chronic stress is a major risk factor in the development of anxiety disorders, and in this specific case, we're talking about post-traumatic stress disorder, or PTSD, which is characterized by increased anxiety and arousal, intrusive thoughts, and avoidance following trauma. Typically, our bodies can easily recover from acute bouts of stress, both positive and negative, while chronic stress causes dysregulation within the hypothalamic pituitary adrenal, or APA axis. Development of these anxiety disorders increases with trauma load across one's lifetime. However, these alterations become even more detrimental when the trauma occurs during the extremely vulnerable adolescent time period. Not only does this chronic stress lead to irreparable damage within the brain's neurocircuitry, it also causes changes in the body's inflammatory response to an immune challenge. In this case, the immune challenge presented was an acute dose of LPS, or lipopolysaccharide, an endotoxin found in the membrane of gram-negative bacteria. This trigger was expected to cause an exaggerated immune response, as demonstrated by increased production of cytokines IL-1 beta, which regulates inflammatory cell recruitment, and TNF-alpha, which induces fever and inflammation. So for the methods of this study, both male and female mice were separated into 2 groups: the stressed animals, which were isolate housed, and non-stressed animals, which were pair housed for the entirety of the experiment. At the beginning of adolescence, on postnatal day 37, the stressed mice underwent 15 consecutive days of predatory stress, where they were placed into the cage of a male-long Evans rat for 30 minutes each day. Afterwards, both stressed and non-stressed animals performed the open field task as a measure of anxiety-like behaviors, as measured by time spent in the center or around the periphery of the arena. On postnatal day 58, each animal was given an IP injection of LPS and tissue was collected 2 hours post-injection. In this study, we aimed to determine the extent to which sex impacts the development of anxiety-like behavior and altered peripheral immune response following chronic adolescent stress. So with the open field, decreased exploratory behavior or less time spent within the center of the apparatus would suggest an increase in anxiety-like behavior. In this graph, we see a significant decrease in time spent in the center in both males and females within the stress group when compared to their control counterparts. This would be indicative of an anxiety-like phenotype within the stress group. On the left, we see IL-1 beta levels within the periphery as measured from the plasma. On the right, we see TNF-alpha levels within the periphery. Both baseline and terminal cytokine levels were measured using the MSD VPLEX pro-inflammatory panel, and baseline levels of each cytokine are depicted here by the dotted lines for comparison to the terminal levels. While we did not observe stress-related differences in circulating cytokine levels, we did observe a stress-specific difference in peripheral cytokine levels. While IL-1 beta circulation did not increase following predatory stress exposure in response to an acute inflammatory trigger, as displayed in Figure 1, peripheral levels of TNF-alpha did increase, as pictured in the second figure. The data presented here demonstrate that regardless of sex, chronic adolescent predatory stress induces anxiety-like behaviors as evidenced by a decreased time within the center of the open field and exacerbated peripheral inflammatory reactivity as shown by increased peripheral TNF-alpha levels. Exposure to this chronic stress during the developmental time of adolescence leads to an immediate consequences, including behavioral deficits and inflammation. The observation that chronic adolescent stress causes an increase in TNF-alpha production following LPS in the periphery without causing the same response within the brain suggests that neuronal inflammation is regulated by different mechanics or possibly on a different temporal scale. Additional predatory stress studies are necessary to identify the mechanisms by which stress drives this inflammatory alteration, but these data here highlight the utility of predatory stress for precipitating similar responses within males and females. So I would like to thank my fellow lab members and my PI for their help throughout this project, Black in Neuro for this presentation opportunity, as well as our funding source. Thank you.
Thank you so much, Samaya. Next up, we have another pre-recorded talk from Joyce Stipersky.
First, I would like to thank the organizers. It is a great pleasure to present my work in this event. So today I'm going to talk about how ethanol gestation exposure affects brain endothelial function and the formation of a blood-brain barrier. So ethanol is the psychotropic illicit drug most consumed in the world. It induces depressing effects and has no specific cellular target since it diffuses rapidly and widely for all body tissues. Chronic ethanol intake functionally impairs several organs and systems such as the CNS, and gestational exposure to ethanol induces body and CNS teratogenic effects, characterizing the fetal alcohol syndrome. Children affected with FAS present severe anatomical alterations in the CNS, such as microcephaly and also several cognitive deficits. In the US, FAS surpasses the prevalence of other developmental-related diseases, and recent studies reveal that per year, about 630,000 children are diagnosed with FAS in the world. In the CNS, alcohol exposure impairs neurostem cell proliferation, migration, and differentiation, induces apoptosis, astrocyte function deficits, and neuroinflammation by induction of pro-inflammatory cytokine production by microglia activation. Interaction of brain vasculature with neural cells such as astrocytes is essential to provide nutrients, oxygen, and to control the passage of substances from bloodstream to brain parenchyma, characterizing the main functions of the blood-brain barrier. Until now, little is known about the effect of ethanol exposure on vascular development and BBB establishment. In this context, we asked, how is gestational exposure to ethanol affects blood vessel development and endothelial function in the developing CNS, and also whether endothelial dysfunction could be propagated to neural cells, such as astrocytes. To answer that, we developed a fast model in vivo by administering ethanol to pregnant Swiss Webster mice from gestational day 14 to 19, 1 dose a day. And in this model, we observed typical fast characteristics such as decreased body and brain weight, and also reduction in cortical thickness. And surprisingly, in the cerebral cortex of pups exposed to ethanol, we observed hypervascularization by labeling blood vessels with isolectin B4 and altered distribution of tight junction protein Z01, suggesting that barrier function of the vascular endothelial cells could be impaired in vivo. To better address the effect of ethanol on endothelial function, we cultivated human brain microcapillary endothelial cells and treated with different concentrations of ethanol, and observed that although total cell numbers and viability was not affected, it critically altered BBB-related molecules levels, since it decreased the expression of tight junction Z01 and claudin 5 protein and mRNA levels, and also disorganized the distribution of tight junctions in endothelial cell membrane. Further, glucose transporter GLUT1 levels was also reduced, which was accompanied by decreased glucose uptake activity. In addition, proteomic analysis of the conditioned medium from endothelial cells treated with ethanol revealed altered levels of angiogenesis-related proteins. Most of these proteins are also involved in several other biological processes which are essential to CNS development. This group of results suggests that ethanol exposure, in fact, induces endothelial dysfunction in vivo and in vitro. And next, we asked whether endothelial dysfunction could be propagated to astrocytes. In our FAST model, we observed that an increase in the expression of GFAP, an astrocyte marker, and the association of astrocytes perivascular and fit with blood vessels was also altered, suggesting that an abnormal development of the BBB. In fact, treatment of murine cortical astrocyte cultures with a conditioned medium from endothelial cells previously exposed to ethanol induced the expression of genes involved with BBB function and glial reactivity, such as aquaporin, connexin 43, lipocalin 2, and GFAP bars, shown by the black bars here. And also, in the same context, we observed induction of the expression of pro-inflammatory cytokines such as IL-1 beta, IL-6, and TNF-alpha, and also activation and nuclear translocation of the NF-kappa B transcription factor. Astrocytes treated with a conditioned medium from untreated endothelial cells did not provide the same response. So, these results suggest that endothelial cells exposed to ethanol possibly induces an astrocytic reactive and pro-inflammatory phenotype. In conclusion, we described that teratogenic effects of ethanol on neurovascular interactions in the developing brain promotes hypervascularization, promotes structural and functional deficits in endothelial cells, and changes the astrocyte and vascular association, since endothelial dysfunction induced by ethanol could be propagated to astrocytes that in turn switch to a reactive and pro-inflammatory phenotype. I would like to thank people in the lab, especially two PhD students, Michele Siqueira and Fernanda Brito, which are directly involved in the fetal alcohol syndrome project, and also want to thank Dr. Flavia Gomes for our collaboration in this project. Thank you.
Thank you so much. Now we'll go ahead and take questions for those four speakers. I also have a quick update. After this Q&A, we'll have one more presentation that we're really excited about. So stay tuned for that. Oh, so if you want to go ahead and drop your questions in the chat for Jamal, Freedom, Samaya, or Joyce, that would be great. First up, we have a question for Jamal. The question is, do you think higher level regions are more sensitive to reward prediction errors versus the auditory cortex?
Can you hear me?
Yes.
Okay. I'm actually not sure. In my study, I highlighted precuneus and angular gyrus, but we VMPFC, which I think has been shown to respond to reward related to music. But I don't know enough about reward prediction errors or anything in Petunias and Angular gyrus. To answer the question, sorry.
Okay, great, thank you. Next up, we had another question for Jamal. They said, very cool research. Apologies if I missed this during your talk, but is the way music is chunked specific to genre? And did you find any differences in your results regarding classical versus jazz?
The chunking, I haven't looked at whether that differs between the genres, but I have done some genre analyses. Like I've been able to do decoding or classification of activity between genres. But yeah, there's a lot of other stuff I would love to do with the genre stuff, but I haven't gotten around to it yet. So yeah.
Great, thank you. Our next question is for Freedom. This person said, very intriguing, and they asked about the homology between pup rearing and compassion. If I think about an evil person who nonetheless cares for her children, is this person applying compassion selectively, or is child rearing potentially different from compassion?
Well, yeah, I like that question because with optogenetics, what we're doing really is trying to separate, the neurons that we're activating. So if we know which neurons are for compassion or for child rearing or whatever, so we can limit our analysis. So somebody could actually have like the structural aspect of child rearing and doing all the right things, but the brain activity is going to tell you if, that's just, I'm just doing my job, or I really, really care and I'm compassionate about that child. And I think that is why optogenetics is really more effective than electrophysiological and other ways of investigating brain action and activity.
Great, thank you, Freedom. This next question is for Samaya. Does chronic rat exposure lead to other stressors, like lack of sleep? And might these contribute to the anxiety-like phenotypes that you see? Oh, you're muted, Samaya.
Sorry. So we have not specifically looked at any specific changes from the rat exposure in changes in sleep, but we have noticed different changes in learning and memory, cognition, and things of the such.
Great, thank you. I have another question I believe this is for Freedom. This person said, nice presentation, and asked, do you think empathy has the same neurological basis for compassion? So kind of similar to before, but curious if those two things have a different neurological basis.
Yeah, that's a good question. I think you have to now look at the distinction between empathy and compassion itself. They are actually distinct. Empathy is really putting yourself in somebody's shoes. It's like literally, seeing their point of view, and there's no action to it. I don't have to do anything. I'm just acknowledging whatever you're going through. It could be grief, it could be, whatever it is. Whereas compassion, on one hand, has an action to it. And so if you look at the neurological basis, compassion has that active aspect to say, hey, I see that this person is suffering, and I'm going to do this particular act to alleviate their suffering. So they are definitely pathways that are shared, but they are also different neurological pathways.
Great, thank you. This last question is for Jamal. Why did you choose classical and jazz music to use in your methodology?
For a few reasons, actually. One was because we were trying to collect naturalistic stimuli for the music study, right? But we also wanted sort of theoretically well-defined music. And so that happens to be the case for a lot of classical and jazz music. And then we also were hoping to look at how higher order brain regions would schematically represent each of the genres, and whether you'd see different schema representations for classical versus jazz. And I'm still working on those analyses. So that's why, yeah.
Great, thank you. Again, I want to thank everyone for being here today, especially during these Somewhat tumultuous times. I want to thank all of our speakers, all of our judges, and a special shout out to our conference sponsors. This concludes our first virtual conference, during which we held several events. We started last week with a collaboration with NeuroMatch, and then Saturday began our official conference programming. We held a social, we had a recruitment event, and on Sunday, we had several professional development workshops and panels. Then we brought you 2 days full of science presentations during what we know was a somewhat difficult time, like I said, but I'm hoping this could be a bit of a bright spot for you, provide some distraction, and hopefully give you some inspiration for the future. If you could stay tuned for our post-event survey, we definitely want to get your feedback. We're a young organization, and we are always looking for feedback on how we can best grow and evolve so we can best serve you in our community. I would also like to thank both of our keynote speakers, Dr. Gina Poe and Dr. Nee Addy, for their fantastic keynotes that I'm sure will be very inspiring to everyone watching. And last but not least, I'd like to thank our Black and Neuro team, especially everyone on the conference planning committee who worked tirelessly to put this event together for you. And the biggest thanks of all goes to our conference co-chair, Monica Javidnia, who tirelessly went through all of your presentations and all of your abstract submissions and all of your judging forms, and really put together this entire the entire 2 days of scientific session. So we definitely couldn't have done it without her. So we thank her so much. So with that, I'd like to, wish you all a great rest of your day, and thank you again for being here.